In this microlecture, we will focus on the process of mitosis and cytokinesis. Whether this material is new or review, please use this microlecture and your textbook to help you understand the purpose, steps, and components of mitosis and cytokinesis. We will use this information over the next week as we discuss the other stages of the cell cycle and how these stages are regulated. As we mentioned in class, the cell cycle refers to the ordered series of steps that takes place in a eukaryotic cell leading to cell division and duplication. The cell cycle can be divided into the M phase and interphase. We will discuss the events that occur during interphase later this week. M phase includes the processes of mitosis or meiosis, which is the division of the nucleus, and cytokinesis, which is the division of the cytoplasm. We will discuss meiosis in greater detail during class next week. In this microlecture, we will focus on the process of mitosis and cytokinesis. Humans have 46 chromosomes in the nucleus of each somatic cell. During interphase of the cell cycle, each chromosome is replicated through the process of DNA replication that we discussed previously in class. As a result of DNA replication, each somatic cell nucleus contains 46 chromosomes that are made of 2 genetically identical sister chromatids. The sister chromatids are connected at the centromere. They are also connected along their entire length by cohesins. During the process of mitosis, the sister chromatids of each chromosome will be separated, thereby forming 2 copies of each chromosome. 1 copy of each chromosome will be incorporated into the nucleus of each daughter cell. Thus, after cytokinesis, the 2 daughter cells will each have 46 chromosomes in their nuclei. Altogether, the process of mitosis, followed by cytokinesis, results in 2 daughter cells that are genetically identical to the parent and contain the same number of chromosomes as the parent cell. The process of mitosis can be divided into 5 subphases that are called prophase, prometaphase, metaphase, anaphase, and telophase. We will examine them all in turn. Before we start discussing the steps of mitosis, let's take a moment to integrate information covered throughout this course by answering a thought question. As seen in the electron micrographs on this slide, during interphase the chromosomes are uncondensed. What general processes occur during interphase that would benefit from this chromosome structure? Think about this question for a minute, then check the slide at the end of the microlecture for some answers. All right, now let's focus on the first step of mitosis called prophase. The uncondensed form of chromosomes present during interphase is not conducive to the organized movement of chromosomes that must occur during mitosis. Thus, during prophase, the chromosomes condense into compact structures. This difference in chromosome structure can be seen in the fluorescent micrographs shown on this slide, in which the chromosomes are stained blue. Notice how the entire nucleus looks blue in the interphase cell, while discrete chromosomes can be seen in the prophase cell. The other major event that occurs during prophase is the formation of the spindle apparatus. In the fluorescent micrographs shown on this slide, the spindle apparatus looks green. The spindle apparatus is essential for moving chromosomes during early mitosis and separating sister chromatids during late mitosis. The spindle apparatus is made up of microtubules. As discussed in class last week, microtubules are one type of cytoskeletal element. During mitosis, the microtubules of the spindle apparatus are responsible for moving chromosomes around the cell. The minus end of the microtubules originates at the centrosome, which is a specialized type of microtubule organizing center present in animal cells. There are two centrosomes in the cell associated with the spindle apparatus. These centrosomes will ultimately be located at the poles of the cell and will define the poles of the spindle apparatus. During prophase of mitosis, the spindle starts to form around the chromosomes by moving the two centrosomes to the opposite sides or poles of the cell. Thus, microtubules that extend from each spindle pole and overlap with one another are called polar microtubules. The second step in mitosis is called prometaphase. There are two major events that occur during prometaphase: the disintegration of the nuclear envelope and the movement of the chromosomes towards the center of the cell. As mentioned earlier, the spindle apparatus is essential for chromosome movement. A second type of microtubule, called kinetochore microtubules, are important for this process during prometaphase. Kinetochore microtubules originate in the centrosome like polar microtubules. However, instead of overlapping with other polar microtubules, kinetochore microtubules attach to chromosomes at specialized structures called the kinetochore. Kinetochores are assembled at the centromere, and each sister chromatid has its own kinetochore. Once kinetochore microtubules are attached to each sister chromatid at the kinetochore, the chromosomes start to move to the center of the cell due to the continued movement of each centrosome to the opposite poles of the cell. The third step in mitosis is called metaphase. Metaphase begins once the chromosomes have moved to the middle of the spindle apparatus and thus the middle of the cell. At this point, not only are the chromosomes at the middle of the spindle apparatus, they are also aligned such that one sister chromatid of each chromosome is oriented towards one of the poles of the spindle. The line at which this alignment occurs is called the metaphase plate. This event concludes the formation of the spindle apparatus. The complete spindle apparatus thus consists of polar microtubules, which overlap in the middle of the cell at the metaphase plate, and kinetochore microtubules that associate with each sister chromatid at the kinetochore. Proper alignment of the chromosomes at the metaphase plate depends on the equal amount of tension placed on each sister chromatid by their associated kinetochore microtubule and the constant growth and shortening of these kinetochore microtubules to maintain this tension. The fourth stage of mitosis is called anaphase. During anaphase, the cohesin proteins that hold each pair of sister chromatids together split. This ends the tug of war between the kinetochore microtubules associated with each pair of sister chromatids and gives rise to two unattached sister chromatids, which are now called independent chromosomes. The newly independent chromosomes move towards the opposite poles of the cell due to the shortening of the kinetochore microtubules, which are disassembled at the microtubule plus end associated with the kinetochore. In addition, motor proteins associated with the overlapping polar microtubules push the two poles of the cell further apart. Anaphase is completed when each set of chromosomes has reached the opposite poles of the cell. Let's pause for another moment to reflect on the important role that kinetochore microtubules play during mitosis by answering the following thought question. What would happen if a kinetochore microtubule did not properly attach to the kinetochore of a sister chromatid? Think about this question for a minute, then check the slide at the end of the microlecture for some answers. The final step of mitosis is called telophase. During telophase, the nuclear envelope reforms around each set of chromosomes, and the chromosomes start to decondense. Thus, Mitosis ends when a single, elongated cell contains two independent nuclei. Mitosis is usually immediately followed by the division of the cytoplasm, called cytokinesis. Cytokinesis ensures that the cytoplasmic contents, including organelles, are distributed to both daughter cells. In many eukaryotic cells, including animal cells, cytokinesis begins with the formation of a cleavage furrow at the center of the cell. Cleavage furrow formation and subsequent plasma membrane shrinkage requires another cytoskeletal element called microfilaments. As discussed in class last week, microfilaments are composed of actin monomers. Cleavage furrow formation occurs due to the formation of a ring of actin filaments just inside the plasma membrane at the point that used to be the metaphase plate. The motor protein myosin that associates with actin filaments then uses ATP to contract in a way that causes the actin filaments to slide. Continual myosin movement causes the actin ring to shrink and tighten. Because the actin ring is associated with the plasma membrane, this causes the plasma membrane to pinch inward until it ultimately pinches in two. Thus, at the end of mitosis and cytokinesis, two daughter cells have formed that have the same number of chromosomes as the parent cell and that are genetically identical to the parent cell. You are now ready to put together what you know about mitosis and cytokinesis to think integratively about the cell cycle and the regulation of the cell cycle. What step or steps in mitosis and cytokinesis should be regulated in order to ensure that these processes proceed properly? We will apply these concepts and address this question in class this week as we continue to discuss the cell cycle and its regulation. Answers to the thought questions are on the next slide. As always, please be certain to get in touch with your instructors if you have any questions about this microlecture or any aspect of the course.